Hello everybody. Can you hear me?
Yes, ma'am.
Okay, thank
Good
you.
afternoon, ma'am.
Good afternoon. Okay, so now we will know about the venous drainage of the central nervous system. Already you have been taught about that arterial system, isn't it? Arterial, the arteries of the brain. Already you have read, isn't it?
Yes, ma'am.
Okay, so today venous drainage, but it is a very small lecture. So I have I will take another lecture along with it. That is the development of the eyeball, because you wanted to have that lecture also. So I will take the development of the eye. So I will go first. The venous drainage of the central nervous system. Okay, so venous drainage, you can see the brain. The brain has what are the parts of the brain? The cerebrum and the cerebellum, as well as cerebrum, cerebellum, and then what is there? The brain stem. So these three parts, they will form the brain, and there is also the central nervous system. It is of the brain and the spinal cord. So below the brain stem is the spinal cord. Now these veins of the brain and the central nervous system, particularly in the brain, the veins, they will drain into the dural venous sinuses. That has been already been taught. Okay, so these are the dural venous sinuses. Like this is the superior superior cerebral sinus or the sagittal sinus. Then here is the transverse sinus. Then the sigmoid sinus. Then ultimately it will form the internal jugular vein. So all the veins of the brain, they will drain into ultimately into the internal jugular vein. In the spinal cord, these veins, they will drain into two plexus. That is, the one is known as the internal vertebral venous plexus, and another is outside the vertebra, that is external vertebral venous plexus. Now the veins of the brain and the spinal cord, they are very thin walled. They don't have any muscles. Okay, smooth muscles are not there. So and they are valveless also. They are not. Their valves are not present. Usually in outside the central nervous system, the veins they have valves. But here, in case of central nervous system, there is no valve inside the veins. Then we will first start with the venous drainage of the spinal cord. So in that venous drainage in the spinal cord, there are 6 veins. The 2 veins are present in the one in the anterior median fissure and another in the posterior median sulcus. So that is the anterior median spinal vein. This is the posterior median spinal vein. Similarly, just behind the anterior root and behind the posterior root, there will be the 2 anterolateral veins and the 2 posterolateral veins. Now these veins, they will drain into the internal vertebral venous plexus, that is inside the vertebral foramen. That is the anterior part of the internal vertebral venous plexus, that is the posterior part. The anterior part is more prominent. Okay, and this is the outside the vertebra. There is another plexus, that is external vertebral venous plexus, and it is present in front of the vertebral body and also in the behind the vertebra, that is behind the transverse process, behind the lamina, behind the spinous process, by the side of the spinous process, etc. Now the internal vertebral venous plexus and the external vertebral venous plexus, they are connected by the basivertebral vein. Basivertebral vein, these are the basivertebral vein. So these veins, they connect the internal and the external vertebral venous plexus. All these veins are very thin walled and they have no valves. Okay. Also, along with these veins, there are some radicular veins are present. So what are the radicular veins? These veins pass along the anterior root and the posterior root of the spinal nerve, and they drain into the segmental vein outside. Okay, the external vertebral venous plexus also ultimately drain into the segmental vein. Now here you can see that this is the these are the radicular vein. These are known as the radicular spinal medullary vein, radicular spinal medullary vein. So there is anterior radicular spinal medullary vein, posterior radicular spinal medullary vein. So they will drain into the segmental vein. In the cervical region, they will drain into the deep cervical vein, vertebral vein, or in the jugular vein. In the thoracic segment, they will drain into the azygous vein or into the hemiazygous vein. In the lumbar region, they will drain into the lumbar vein. Also, the external vertebral venous plexus also drain into the segmental vein. Now, one of this radicular vein, anterior radicular vein, is very thick. It is very dilated, and that is known as the great anterior radiculomedullary vein. Great anterior radiculomedullary vein, or you can told this that is the GARV, GARV. Okay, so great anterior radicular medullary vein. Now it is present in the lower part of the thoracolumbar segment. In that segment, in all the 11th or 12th thoracic segment, that is there one artery is present, that is known as the arteria radicularis magna of Adam Quiz. Have you heard that name? Arteria radicularis magna of Adam Quiz?
No, ma'am.
Right? Arteries, you have read about the arteries, na? You don't know arteria radicularis magna. That is because there are the anterior spinal artery and the posterior spinal artery, and the lower part of the spinal cord, they are very thin. So there is one artery comes from outside, that is the radicular artery, that is known as, that is very thick artery or dilated artery, that is known as the arteria radicularis magna, which supply the lower part of the spinal cord. So in the blood supply of brain, when you will spinal cord, when you will read about that, you will get that name, the arteria radicularis magna of Adam Quiz. So it is very important. Similarly, there is a vein, though it may not accompanying the arteria radicularis magna, but it is present in that same level, and sometimes it may be thought that it is the artery, it is not the vein, but actually it is a vein. Okay, and these veins have no valves, and they do not follow the arterial. In our body, the veins, they accompany the arteries, but in the central nervous system, the veins, they do not accompany the arteries. Okay. Now, the vertebral venous plexus I have already told you. Now, what is Batson's plexus? That is the Batson's plexus. It is the external vertebral venous plexus. Actually, it is the external vertebral venous plexus. And I have told you that external vertebral venous plexus, in the cervical region, it will drain into the deep cervical vein, vertebral vein. In the thoracic region, it will drain into the azygous and the hemiazygous veins. In the lumbar region, it will drain into the lumbar vein, and this lumbar vein ultimately communicating with the pelvic veins. Okay, so when there is any infection or when there is any carcinoma in this region, so this the carcinomatous cells or the cancer cells or the infected materials can pass through these veins into the Batson's venous plexus, and from there it may cause the cancer of the vertebrae or the cancer of the brain. Okay, so that is the applied importance of the Batson's plexus. And also the infection may cause osteoarthritis in the vertebrae. If there is any infection, that will pass through this Batson's venous plexus from the pelvic region and may cause osteoarthritis in the vertebra. So in that case, the pelvic infection, if it is corrected by any specific antibiotic, that antibiotic will also correct the osteoarthritis in the vertebra. Okay. Now we are coming to the venous drainage of the brain. Now in this venous drainage of the vein, there are superficial veins and the deep veins. So superficial veins, they can be divided into three types. One is the superior cerebral vein, middle cerebral vein, and the inferior cerebral vein. So the superior cerebral vein, they are present in the superolateral surface as well as on the medial surface. This is the medial surface, as well as on the inferior surface. So the superior cerebral, not on the inferior surface, in the superolateral surface and on the medial surface. This is medial surface. Okay. So these are the superior cerebral veins, you can see here. So superficial venous drainage is the superficial vein, deep vein, and also the specific veins that will drain the brain stem and the cerebellum. Superficial veins, I am telling you that this is superior, middle, and inferior. Deep veins, there are many. Later I will come to that. And there are veins for the brain stem. Brain stem is made up of medulla, pons, and the midbrain. And cerebellum is behind the brain stem. So they are also the parts of the brain. Okay. Now the superficial vein, I have told you that is there are superior veins, that is the superolateral surface. So these are the superior vein which are draining into the that is the superior sagittal sinus. Now here you can see the blood is flowing from in the superior sagittal sinus from the anterior aspect to the posterior aspect, whereas the superior cerebral vein which are draining into the superior sagittal sinus, their flow is from the posterior side to anterior side, just on the opposite side. So why it is like that? So in that, so there are two types of flow. One is from the anterior to posterior, another from posterior to anterior. And that will prevent the collapse of the veins, because these veins, they do not have any smooth muscles. So they are very soft. So to prevent the collapse in case of increased intracranial pressure, to prevent this collapse of the veins, the flows are different. Okay. So they are draining here, and now you can see they are draining on the superior sagittal sinus. Okay, that is the superior cerebral vein. Now on the medial surface also they are present. They are here. They are present on the superior sagittal sinus. Okay. Now you come to the medial cerebral vein. So medial cerebral vein, it is present, that is also maybe superficial and deep. So the superficial one is present in the lateral sulcus. So if this is the superolateral surface, there is a deep sulcus is present here, and that is known as the lateral sulcus, stem of the lateral sulcus. So in the stem of the lateral sulcus, this superficial middle cerebral vein is present. The deep middle cerebral vein is also present in that area, but to a deeper aspect. The deep veins, they drain the deeper structure, like the deep cortical, deep medullary substance, white matter, or and also the nuclei which are present in the deep part of the cerebral hemisphere, like they are known as the corpus striatum or the basal ganglia. The deep veins also drain the thalamus. Okay, the deep veins, they also drain the striate area, the choroid area, choroid plexus. So there the deep veins are formed. So this, we are talking about the superficial veins. So superficial veins, superior veins, I have already told you. Now I am telling you about the superficial middle cerebral vein. And this superficial middle cerebral vein, it is connected with the superior sagittal sinus by means of the superior anastomotic vein, and with the transverse sinus by means of inferior anastomotic vein. Can you see that? Two veins. This is the superior anastomotic vein and this is the inferior anastomotic vein. And the superficial cerebral, middle cerebral vein may drain into the superior sagittal sinus or into the transverse sinus through the inferior anastomotic vein. Okay. Now you come to the inferior cerebral vein. So the inferior cerebral vein, they are also present on the superolateral surface on the medial surface as well as on the inferior surface. So if we see, these are the inferior cerebral vein. So where they are draining? They are draining into the superficial middle cerebral vein. This is the inferior vein. They are draining into the transverse sinus. On the medial surface, they will drain into the straight sinus, okay, or into the transverse sinus. And the inferior surface also, the veins are present. These are known as the inferior cerebral vein. Now, here you can see this is the inferior surface of the brain, okay. This is the frontal part. This part is known as the orbital surface of the brain. So from there, these are coming to the cavernous sinus. From the cavernous sinus to the superior petrosal and inferior petrosal sinus, they will drain into the transverse sinus. Ultimately, the transverse sinus will go to the sigmoid sinus and that will form the internal jugular vein. Okay, so these are the venous drainage. Now come to the deep cerebral vein. The deep cerebral vein, there are many deep cerebral vein. One is the thalamostriate vein, another is the internal cerebral vein here, then the great cerebral vein, then the basal vein, and also the deep middle cerebral vein. So these are the deep brains. Now thalamostriate veins, its name suggests that it is draining the thalamus as well as the basal ganglia. The striate area is also known as the basal ganglia. So there are various deep nuclei are present in the white substance. So these nuclei are known as the basal nuclei. So from there the thalamostriate veins will come and it will also drain the septal area. And ultimately, the two thalamostriate veins, they will join with one another in front of the thalamus. There is a foramen. That foramen is known as the foramen of Monro. Now, through that foramen of Monro, the lateral ventricle and the third ventricle, they communicate with each other. In that region, the two internal cerebral veins are formed by the union of the thalamostriate vein of each side. Okay, so this is the internal cerebral vein. There are two internal cerebral veins. Now these two internal cerebral veins ultimately join with each other in the posterior part of the corpus callosum, where it will form the great cerebral vein of Galen. And that great cerebral vein, it passes deep to the splenium of the corpus callosum and drain into the straight sinus. Now there is another vein which is formed in the region of the anterior perforated substance, and I will show the anterior perforated substance where it is. Now here you can see the basal vein. They are of two sides also. Here they have cut it. So on that side they are showing only on the right side. The left sided basal vein is not shown. The right sided basal vein, you can see that it is going behind the brainstem. So there are two veins. They will go on each side of the brainstem, that is the midbrain, and ultimately they join with each other and they will drain into the great cerebral vein. Okay, they will drain into the great cerebral vein. So how this basal vein is formed? Now, if you see the inferior surface of the brain, you will see that there is the optic chiasma in this region. By the side of the optic chiasma, there is an area which is perforated. This perforation is by the deep arteries which are going through this anterior perforated substance. So in this area, the anterior cerebral vein, which is present just above the corpus callosum, it will come, and also this is the stem of the lateral sulcus, which is continuous on the lateral superolateral aspect also. That is the middle cerebral vein, and another vein, that is the inferior striate vein. So these three veins, they will join together, the anterior cerebral, middle cerebral and the inferior striate vein, they will join together and they form the basal vein. So the basal vein on both sides, they surrounds the, this is the midbrain area, they have cut it. So here you can see on both sides of the midbrain they are coming and they will ultimately drain into the great cerebral vein, which is continuous with the straight sinus. Okay, so these are the deep veins. Now, what is that? Sorry. The anterior cerebral vein. So this is the anterior cerebral vein. Okay, so this is the here is the optic chiasma. So in this region there is the what structure is present, that is the anterior perforated substance. So the anterior cerebral vein, middle cerebral vein and the inferior cerebral vein, they will form the basal vein. Okay. Now the veins draining the brain stem. So brain stem, I have told you, that is the midbrain in this region. This is the pons and this is the medulla. So in the midbrain, the basal vein is there. So basal vein, it drains the midbrain. Also there is pontomesencephalic sulcus. Here the veins are present in that region at the junction of the midbrain and the pons. So that is the pontomesencephalic sulcus, they are present. In the pons there is the median pontine vein and the lateral pontine vein, and they are connected with each other with the transverse pontine vein. Similarly in the medulla there is median medullary vein, lateral medullary vein, and they are connected with each other by the transverse medullary vein. Okay, so these are the venous drainage of the brain stem. What about the cerebellum? The cerebellum has two surfaces. One is the superior surface, another is the inferior surface. Here you can see the superior surface. Okay, and here they have shown both the superior surface and the inferior surface. Here which sinus is this? This is the transverse sinus. So from the superior surface, these veins, they will drain into the transverse sinus. Even from the inferior surface, they will drain into the transverse sinus. What is this? This is the straight sinus. So they will also drain into the straight sinus. Here there is the superior petrosal sinus. So superior cerebellar vein, they will drain into the petrosal sinus, into the transverse sinus, into the straight sinus, and ultimately they will go to the sigmoid sinus. So the venous drainage is complete. Is it clear? You have to read it. Okay?
Yes, ma'am.
And you have to remember it also. There are various names. These names are not very much familiar to you. But if you read, then ultimately you can get those names. Okay? Now come to the development of the eyeball. So when the eyeball is developed, that is from the 3rd week to 10th week intrauterine life. And there are sources. These sources are they are developed from the neuroectoderm, from the surface ectoderm, and also from the mesenchyme. Here I have written the mesenchyme invaded by the neural crest cells. Not only the neural crest cells, they also invade the surface ectoderm, they also invade the neuroectoderm. So the neural crest cells also takes an important part for the development of the eyeball. So the neuroectoderm, surface ectoderm, mesenchyme, and the neural crest cells. Okay. Now the development of the central nervous system, you know, there are brain vesicles. 3 brain vesicles are there, and spinal cord from the caudal part of the neural tube it is developing. The 3 brain vesicles are the prosencephalon, the mesencephalon, and the rhombencephalon. And the prosencephalon again divided into telencephalon, diencephalon, mesencephalon remains same, that is the mesencephalon, and the rhombencephalon is divided into metencephalon and myelencephalon. From the telencephalon, the cerebral hemisphere is developed, and there are 2 cerebral vesicles, and the lateral ventricles are formed there. From the diencephalon, the thalamus is developed. The thalamus is developed from the diencephalon. Midbrain is developed from the mesencephalon. Metencephalon, the pons and cerebellum, and the myelencephalon, the medulla oblongata. And this the colored area, that is the wall of the brain, and inside the white area, that is the ventricle or the canal or the cavities of the brain vesicle. So here two lateral ventricle will be formed. Inside the diencephalon, the third ventricle will be formed. Inside the mesencephalon, the cerebral aqueduct will be formed. In the rhombencephalon, the fourth ventricle is the cavity. Okay, now from the diencephalon, that red area, the two optic vesicles that is developed from the neuroectoderm, and it is going towards the surface ectoderm. Okay, now here this is the optic vesicles from the diencephalon, and it is going towards the surface ectoderm. As it touches the surface ectoderm, the surface ectoderm becomes thickened and it forms the lens placode. When there is the surface ectoderm is thickened, then in that region that will form the lens placode. And this optic vesicle ectoderm, that is the neuroectoderm and the surface ectoderm, they will induce each other. As they have induced each other, the lens vesicle will invaginate, and the optic vesicle, optic vesicle will also invaginate in that area. And ultimately what will happen, the lens, it has touched, the optic vesicle has touched the lens placode, that is the surface ectoderm, to form the thickening of the surface ectoderm to form the lens placode. And when there is induction between them, some genetic factors they will be expressed and they will cause induction, and due to this induction the lens placode becomes invaginated and it will form the lens vesicle, and it will be separated from the invaginated optic vesicle, which will later on form the neural part of the retina. So that will form the neural part of the retina. Now the lens vesicle ultimately, that is the invaginated lens vesicle, they will join with each other to form the complete lens vesicle. Later on the lens will be formed, I will come later. Now come to the optic vesicle. The optic vesicle also invaginated, and now the optic vesicle is known as the optic cup. Now optic cup has two layers, the inner layer and the outer layer. The inner layer, that is in contact with the lens vesicle, and it is now separated, so that will form the neural part of the retina, and the outer layer, that will form the outermost layer of the retina, that is the pigment epithelium, retinal pigment epithelium. So in between them there is a space that is known as the internal retinal space. Later on the outer layer and inner layer, they joined with each other, and that was forming the retina. Okay, the optic cup will form the retina, that is the outer retinal pigmented epithelium and the inner nuclear retina. Okay. Now this is the optic cup, and there is invagination, and this invagination ventrally it will also extended in the optic stalk. So where is the optic stalk? Here you can see the optic stalk. So this is the optic vesicle, and it is connected with the lower part of the third ventricle by the optic stalk. So this is the optic stalk. So when it is invaginated, this invagination is also present in the optic stalk, and that is known as the optic fissure, choroid fissure. This is known as the choroid fissure. This is the choroid fissure. Okay, so this is here, if you cut the transverse section, you will get, that is the optic cup. This is the outer layer from which the inner retinal layer will be, outer layer, that is the pigmented layer, will be formed, and this is the inner layer from where the nervous layer of the retina will be formed, and this is the optic choroid fissure. Through that choroid fissure, one artery will enter, and that is known as the hyaloid artery. Hyaloid artery. Along with the artery, some amount of loose mesenchyme surrounding the neuroectoderm and the surface ectoderm, there is loose mesenchyme is present, that will also enter along with the hyaloid artery. And hyaloid artery, it will go to the lens and it will supply the developing lens vesicle. Okay. Now come to the retinal development. So I have already told that there is an outer layer of the optic cup and inner layer of the optic cup. The outer layer of the optic cup will form the pigment epithelium, and the inner layer of the optic cup, that will form the neural layer of the retina. Now here, this is, they have shown it like this is the like brain. You have already the development of brain has been told. I don't know whether you have remembered or not. So there is outer marginal layer and the inner mantle layer. So here in the marginal layer usually the fibers are present, but here the layers of rods and cones are formed. The layers of rods and cones are formed from the marginal layer, and from the mantle layer where the other neurons are formed, these are the other neurons of the mantle retina. These are the bipolar cell, amacrine cells and the ganglion cells, and all these layers are formed by 8 months of intrauterine life. That is just before delivery, isn't it? Now, but the layers of rods and cone, they are mature even after birth. So when the baby borns, his or her, that baby's eyes are not very much fixed. Their gauge is not fixed. Okay, so that time their rods and cone cells, they are developing. So when they are developed, then the gauge of the baby will be fixed. So these are now these ganglion cells. Ultimately, the light comes to the layers of the rods and cone, that is the photoreceptor cells, and this information will go to the ganglion cells and it will pass to the brain through the axons of the ganglion cells, and these axons, they will form the optic nerve. Now these optic nerve fibers, they will pass through the optic stalk. Ultimately they will pass below the third ventricle, and that optic stalk is converted into optic nerve. I don't know whether you are understanding or not. Okay, now what happened to the hyaloid artery? The hyaloid artery, it has entered to supply the lens. But when the lens is formed, and along with that mesenchyme also entered in here, and in that mesenchyme the vitreous humor behind the lens is formed. In the mesenchyme the aqueous humor is also formed. So they will supply the lens. At that time, the lens, this hyaloid artery, ultimately it starts to degenerate, and only the most posterior part may remain as the central artery of retina. So the hyaloid artery, it is actually a branch of the ophthalmic artery. It will be, its distal part will be that will be degenerated, and the proximal part will remain as the central artery of the retina. And that central artery, that optic stalk ultimately form the optic nerve. So the hyaloid artery, it passes through the optic, that is through the choroid fissure, through the optic stalk. Therefore, the central artery of the retina, it is inside the optic nerve, in the middle of the optic nerve, it is there. Okay, and now the choroid and sclera, how they are formed. Now this is the optic nerve, and this is the mesenchyme surrounding the optic cup. So mesenchyme surrounding the optic cup will form the choroid and the sclera. The choroid, that is a vascular layer, pigmented vascular layer is the choroid, pigmented vascular layer is the choroid outside the neural layer, and the another layer outside that outside this pigmented layer is the fibrous layer, and that will form the sclera. And this sclera is continuous with the dura mater, and the choroid layer is continuous with the pia mater. So this scleroid, the sclera is continuous with the dura mater surrounding the optic nerve, and the choroid is continuous with the pia mater surrounding the optic nerve. So the optic nerve is surrounded by dura mater and the pia mater. In between them there will be the subarachnoid space. That is the arachnoid matter will be there. Okay. Now the cornea, the sclera, that is this part, that is the fibrous layer is the sclera. Now here the choroid and sclera has formed. Now the cornea, how it is formed? So cornea, we know that it is actually the continuation of the sclera, but this continuation of the sclera, which part of the that is the stroma of the cornea is actually continuous with the sclera. The anterior epithelium of the cornea, it is formed by the surface ectoderm. The anterior epithelium of the cornea is formed by the surface ectoderm, whereas the endothelium, it is also formed by the mesenchyme. So the cornea has three parts. One is the anterior epithelium, one is the stroma of the cornea, another is the posterior endothelium. So anterior epithelium, it is formed by the surface ectoderm, and the stroma of the cornea, and also the this is which part? This is the endothelium. It is formed from the mesoderm. Now between the cornea and the lens, there is also mesenchyme was present, and in that mesenchyme the spaces are present, and in these spaces they coalesce with each other. Ultimately they will form a cavity, and in that cavity this choroid, the anterior part of the choroid, that is and in the anterior part they are thin, and they will form the ciliary body and the iris. Now from this ciliary body the aqueous humor is secreted, and that aqueous humor, it is present in between the cornea and the lens. So this is the aqueous humor. Now in this region, when the cavity will be formed in the region, it will divided the mesoderm into two parts, the anterior part and the posterior part. The anterior part will form the stroma of the cornea, and the posterior part, that will form the iridopupillary membrane. Iridopupillary membrane, that is present in front of the lens. And ultimately this iridopupillary membrane will disappear, and at that time the anterior chamber and the posterior chamber, so this iridopupillary membrane, it forms the anterior chamber and the posterior chamber. So anterior and posterior chamber both are filled up with aqueous humor. When the iridopupillary membrane disappears, then only the anterior chamber and the posterior chamber, they will continuous with each other through the pupil. Okay. Now come to the development of the ciliary body and iris. I have told you, so because all these structures, they are developing simultaneously, they are developing together. Okay, so when the aqueous humor is formed, then at that time the ciliary body and the iris is also formed. So the ciliary body, that is and the iris, they are the anterior continuation of the choroid. So this just deep to the choroid, there is the neural layer of the retina, and that neural layer, that there are 10 layers are present, and these 10 layers are present only in the posterior 5/6 of the eyeball. In the anterior 1/5 part, this retina is very thin, and only the pigment layer is continuous, and one of the nervous layer may be continuous with this pigment layer. Okay, and these two layers of the retina, they will form the part of the ciliary body and the part of the iris. Okay, and there when the ciliary body is formed, there are some foldings are present, and these foldings are known as the ciliary processes, and this is the iris is formed here. So the ciliary body, it is and both the iris, they are formed by the neuroectoderm, that is from the optic cup, that is from the neural layer of the optic cup. They will form the ciliary body and the iris. But also the mesoderm will also form the ciliary body and the iris, that is continuous with the choroid. That is continuous with the choroid. So this is the choroid that will also form the ciliary body and the iris, and also the neuroectoderm that will also takes part in the formation of the ciliary body and the iris. And also the what is that, neural crest cells are also there. Now the muscles of the sphincter pupillae, and that is of the iris, that is the sphincter pupillae and dilated pupillae, they are developed from the neuroectoderm, whereas the muscles of the ciliary body, that is developed from the mesoderm. So there are the source for the ciliary body and the iris, both by the neuroectoderm and the mesoderm. Okay. Then the lens is also developing at the same time. Already you know that the lens vesicle is formed by the invagination of the lens placode, and ultimately they join with each other and they are separated from the surface ectoderm, and that is forming the lens vesicle. That is about that 7th week of intrauterine life. Now then after 7th week, this is the posterior epithelium, this is the anterior epithelium. The posterior epithelium will be elongated and they will form the primary lens fibers. These primary lens fibers, ultimately they will form the nucleus of the lens. And the anterior epithelium, this is the anterior epithelium, that is the equatorial zone. That zone is known as the germinative zone. From this germinative zone, the circumferential lens fibers or the secondary fibers, they will come out and they will form the soft cortical part of the lens. Now initially they were, they are circumferentially present surrounding the primary lens fibers that will form the lens nucleus. But later on, when the lens grows in size, they cannot they cannot encircle the nucleus of the lens, and they are going only up to the poles, that is up to the anterior pole and the posterior pole, where they interdigitate with the other opposite sided fibers. So here that will form the lens suture. Opposite the anterior pole and the posterior pole, you will get the lens suture, because here the secondary fibers, they interdigitate with each other, So there the secondary fibers are present. Now how the extraocular muscles which move the eyeball, how they will be formed. So the extraocular muscles are developed from the pre-otic somite. So what is the otic somite? That is the thickening of the surface ectoderm which will form the ear. So here is the otic somite is present in the region of the rhombencephalon. Okay. So before that there are the somites. Somites are the part of the paraxial mesoderm. So these are known as the pre-otic somite. Behind the pre-otic somite there is the post-otic somite. So pre-otic somite gives rise to the extraocular muscles. So there are 6 extraocular muscles and there are 3 pre-otic somite. From the first pre-otic somite, the 5 muscles are formed, okay, and they are supplied by the oculomotor nerve. And from the second pre-otic somite, only the superior oblique muscles are formed, and that is supplied by the trochlear nerve. From the third pre-otic somite, the lateral rectus is formed, and that is supplied by the abducens nerve. So these pre-otic somites, they are supplied by 3 nerves, the third nerve, fourth nerve and the sixth nerve, and they will supply the extraocular muscles. Post-otic somite gives rise to the tongue muscles, which is supplied by the hypoglossal nerve. Okay, so extraocular muscle. Now, so development of eye, so that is the surface ectoderm, neuroectoderm, mesenchyme, neural crest cell. Surface ectoderm will form the lens. The anterior corneal epithelium will be formed by the surface ectoderm. Eyelids are also formed by the surface ectoderm. Shall I? Given the I have not kept that slide, the eyelids. So eyelids, they are formed by the surface ectoderm. There are two folds. The two folds, they are formed in part of the in front of the cornea. There are two folds. Okay, so here is the eyelid is forming like here. So eyelid is formed in front of the cornea. So this is the cornea. This is the cornea. In front of that there will be the two folds of the surface ectoderm is there, and these two folds are continuous with each other. These two folds, they are unite with each other in front of the anterior pole of the cornea. Only at about 7th month these folds, they will be separated from each other. Now when this in this mesoderm between the surface epithelium and the cornea, there is the mesenchyme is present, and there are some spaces will appear, and they will coalesce with each other, and they will form a conjunctival sac. So the eyelid is covered by the conjunctiva, and also it will cover the it will not cover the cornea. But here they have shown. So in this cornea, this is not the conjunctival epithelium. The conjunctival epithelium will only is present up to the sclera. Here it will not be. Later on it will disappear. So this is the conjunctival sac. This is the conjunctival sac. At the end of the 6th month of intrauterine life, these two eyelids, they are separated from each other. Okay, so eyelids are also formed from the surface ectoderm. Okay, the neuroectoderm, the retina, both the pigmented layer of the epithelium and the neural layer, the iris, part of the ciliary body, the iris muscle, that is the sphincter pupillae, dilated pupillae, they are also developed from the neuroectoderm. Optic nerve is also developed from the neuroectoderm. And mesenchyme, that will form the sclera, the corneal stroma, corneal endothelium, choroid, the muscles of the ciliary body, that is also formed from the mesenchyme, vitreous humor, that is also vitreous humor, the neural crest cells, that will also form the vitreous humor, blood vessels. The neural crest cells which invade the mesenchyme, that will also form the sclera, corneal epithelium, and the choroid. Also they will form the vitreous humor. So neural crest cells that will invade the surface ectoderm, neuroectoderm and mesenchyme. So the surface ectoderm, neuroectoderm, mesenchyme and neural crest cells. Okay, this slide. So this is the regulation. I am not going to that. Now there are some abnormalities of the eye. That may be interesting, isn't it? So all these developmental abnormalities, they are present since birth. Now here this is the congenital coloboma. Coloboma means when there is a cut or there is a gap in the iris or in the ciliary body, in the choroid or even in the optic nerve. That may be acquired also due to trauma or injury, but congenitally it may happen when the choroid fissure will not fuse with each other. The choroid fissure will fuse at about 7th week of intrauterine life, but if it does not fuse, then there may be a gap in the iris or in the ciliary body or in the optic cup. Okay. So that is known as the coloboma, congenital coloboma. Another is the persistent iridiopupillary membrane. So here the iridiopupillary membrane, it disappears normally, but if it does not disappear, then you will find that the thin fibrous stroma is present in front of the pupil. So that is the persistent pupillary membrane. It can be removed by surgery. Okay. And then congenital cataract. Congenital cataract, that is presence of cataract since birth, and it may occur in some infection like in rubella infection, syphilis infection, or in any other infection, toxoplasmosis maybe. So there is congenital cataract. It may in one eye or in the both eye. Okay, so that is the congenital cataract. Congenital aphakia. Aphakia means the absence of lens. Usually we know that after when we develop cataract in old age, then when it is mature we remove the lens, and that condition is when there is no lens, that condition is called as aphakia. But here the lens is not present. So if the optic vesicle and the lens vesicle, they do not come in contact with each other, there will be no development of the eye. So in that case, the lens may not be present. So that is the, or in some infection also, congenital aphakia, it is a rare, very rare condition, but it may be present. And that is, sometimes there is persistent hyaloid artery is present. When there is persistent hyaloid artery is present, then there will be abnormal vision and there will be the congenital cataract, again there will be the cataract. Or sometimes only the posterior part of the artery is present, it is known as Burmeister's papillae. Here nothing will happen, there will be normal vision, only the one papillae will be present on the posterior aspect. But if it is present on the anterior aspect, that is also known as some dot, Mittendorf's dot, here also there will be normal vision, nothing abnormality. But if this anterior segment of the vitreous artery, if it enters into the lens to supply it, then there will be abnormal vision and there will be cataract. Okay. Then the developmental abnormalities, now aniridia, that means the absence of iris. That is also a very rare condition, aniridia. Anophthalmia, that is also very rare condition, anophthalmia or microphthalmia, means no eye or very small eye. That is also due to infection like syphilis, rubella, toxoplasmosis, chlamydia, like that. And that condition, that is cyclopia or the synophthalmia, that is the very severe condition. It is also rare, but if it happens, then the fetus will not survive. Usually it dies within the utero, but if it delivers, then within few hours it may die, or the maximum 1 day. Okay, and what will happen? There will be merging of the midline structures. So in case of the brain, that there are two cerebral hemispheres, these two cerebral hemispheres merge with each other. There will be one cerebral hemisphere, there will be only one vesicle there inside, that is the single lateral ventricle will be there. Now you cannot see the nose, but there is the one proboscis is present, snout is present. So that snout is present above the eye. That is developed from the frontonasal process. That frontonasal process is not developing, the nasal placodes are not formed. So that's why the nose is not formed, but the proboscis is formed. Have you seen any proboscis in any animal? In any animals, have you find this proboscis?
Butterfly.
Butterfly? Butterfly mammal हुआ?
Snail.
Snake? वो mammal नहीं है. Any mammal? Elephant?
Tapirs contains proboscis.
Yes, snout. And the elephant also. Elephant's trunk, isn't it? So that is normal for them, but in human being it is not normal. Okay. And that is the proboscis. Tapir, you have named na tapir. In tapir also there is snout. So these are the developmental abnormalities of eye. And till then, thank you. Okay, bye.
Bye.